Good afternoon, everybody, and welcome to this, the fourth session of our single-use solutions webinar. We have worked with single-use solutions at Thurne for more than 15 years within both powder handling containment applications and liquid applications. We have seen the development over the years. It's a development when it comes to different applications or solutions, but it's also an impressive development when it comes to how much this kind of technology is used in more and more different ways and more different applications. We are happy to host this webinar. We do hope that we can bring you to some new insights and ideas for your own use of single-use solutions. And we do hope that even if we have not been able to invite you to a seminar to touch, feel, hands-on experience of the solutions, we hope that we can bring you enough of that through this platform to make it possible for you to somehow visualize how it could be used in your own everyday business. Session of the day will focus on single-use clarification of biological solutions. Our presenter or spokesperson through this presentation will be Bettina Ledergerber from Filtrox. She's a global application engineer for life science at Filtrox. She's been working there for about 2-3 years, and she is responsible for support of filtration trials. So whenever or if any of you would go ahead to make trials, to try out different solutions in small scale to be scaled up, most probably Bettina will be one of the persons to support us with that. Myself, I am Mattias Persson. I'm the moderator of this session. I'm a chemical engineer by background. I worked a few years at AstraZeneca before I joined Thurne. And here I've been more than 15 years working as a sales engineer, section manager, sales manager, and now head of sales for Scandinavian market. If you would have further questions, want to discuss further after this presentation, please feel free to contact your local Thurne representative. It would be myself for Sweden, Norway. It would be Rolf for the Finnish market. It's Rafi in Denmark, and for the Baltic states, it's Andris. Over the word to Bettina Ledergerber from Filtrox.
Thank you very much, Mattias. So I will share my presentation with you. I hope you can see it. And I will talk about the new approach of single-use clarification of biological solutions, as Mattias already told you. First of all, I want to start with a few facts about the company Filtrox. The company foundation was in 1938. We have today now approximately 250 employees worldwide, with 4 production sites and 8 sales offices. And regarding our product, the filter media, we have approximately 5,800 variations. We are close because, as I said to you, we have 8 sales offices, which are worldwide, as you can see here. I am currently in Switzerland, so in St. Gallen, here's the headquarters. We have a sales office here and also a manufacturing site. The other manufacturing sites are in Barnoldswick, so this is in the north from Manchester. Another one is in Czech Republic, and the 4th is in Mexico City. We mainly produce filter sheets, which can then be inserted in sheet filters, so plate and frame filters. But the sheets can also be manufactured as lenticular modules, which can then be inserted in a module housing, so a closed system. We also have cartridges available, which contains sometimes a membrane in it, which can then also be inserted in a cartridge housing, which, as you can see on the bottom, looks quite different, quite similar, sorry, to the module housing. We also have the laser pointer here. We have the single-use series, and I will come back to that just in a second. And we do have some test kits. So this is for scale-up solutions. So with these test kits, you can do some filtration trials in your lab. Then you can scale it up to the production scale. We also do have this filter case. So a whole case which contains everything you need for a lab trial. We also have an all-around assistance. So we do some trainings and seminars, testing services, validation support, and application consulting. First of all, what are the trends in the biomanufacturing? So often the goal is to reduce one production step. And when you can achieve this, sometimes this results in cost reduction. Due to the biological process, we need a robust and flexible technology, fast and reliable, an easy scalable and duplicatable process, and last but not least, particles or impurities must be removed. Solutions for the midstream process. As you may know, you can apply a centrifuge step, you can do a separator step, tangential flow filtration, sedimentation, or acoustic wave. All of these 5 process steps are nice, but they have one main disadvantage: they don't have a zeta potential. So what do we offer as a solution for a midstream process? We offer depth filtration sheets. So the advantages of them is they are easy to use. It is a well-known technology and easy scalable and widely scalable, easy to development and control. More or less no shear stress. Single-use options are available. But the capacity is not high enough. Therefore, we recommend alluvial filtration, which has the same advantages as the depth filtration, but the capacity is higher. Once again, I talk about the midstream process. We like to call it midstream because sometimes it's part of upstream, sometimes part of the downstream, so we call it midstream. Here we have a centrifuge step and a depth filtration step, and with our single-use BioSD system, you can reduce one step, so the centrifuge step, and you can do both in one. So now I want to say something about the functional principle of filtration itself. First of all, surface filtration, as you may know, the particles are retained on the surface, and everything which is smaller than a certain retention rate, the particles going through the filter. The depth filter sheet is quite thicker, so particles are retained also at the surface, but also within the filter sheet by size exclusion. And the yellow squares here at the bottom indicate some particles which are retained by adsorption capacities or effects. And the alluvial filtration, here we add some filter aid to the unfiltered liquid, which is indicated by the gray dots. So our particles, like the cells or cell fragments, will be retained within the filter cake, and the filter sheet or the surface is constantly renewed. So yeah, so the capacity is much higher. We also have some particles which will be retained within the filter sheet, of course. Yeah. What does alluvial filtration mean? So as I said, you have a much higher capacity also for a high particle load, so especially when you work with high cell densities. It is an established technology. It is already used in plasma fractionation, and for long-term, we have long-term experience in the beer filtration. And so far, it was not available as a single-use solution. I want to say some words about the filter aid. We are using Cell Pure, which is an Animaris brand, and it is pharma grade. Another word is diatomaceous earth or kieselguhr. It is inorganic filter aid made yeah, it is mineralized organisms, so the skeleton of diatoms. And from the cell pure, there are 4 grades available. And the difference is in the permeability and the solids to be removed. On this picture, you can see, for example, here, a very nice particle, DE particle. And as you can see, it consists of many little pores, and the pores itself consists again of many little pores. So the liquid is going through the filter cake, and the particles will be retained. The function of the DE, it also helps to clarify your liquid, so not only the sheet itself. So that's why it is very important to use the right DE, so that you have to check in the lab scale which DE grade or type will be the best for you, your process. It avoids clogging of depth filter sheets, and it can remove other impurities like HCP, DNA, etc. And it can do all at the same time. There is one disadvantage, you may know. The powdery material has to get into the bioreactor, which is not very healthy, and that's why you need a powder transfer bag. In this graph, you can see very nice the difference between a filtration with DE and the one without DE. So the blue dots indicates a filtration without DE. So as you can see, the pressure is going up very fast and exponential. And the same filtration with DE, the pressure is going up as well, of course, but linear, and you can filter much more. This is a process comparison. So if you have a large volume, you can add a tangential flow filtration, for example, as a primary clarification. After that, you can apply a depth filtration, or you do it with a centrifugation step at the beginning and then a depth filtration before you go further to the other downstream process steps. And for small volumes, you can add 2 depth filtration steps or acoustic wave and depth filtration. With our system, with the BioSD system, you can do both steps, so primary and secondary clarification in one with the alluvial filtration. So now I come to our range, and I will start in a small scale, the 2-inch capsule. It is mainly used for proof of technology and, of course, for the evaluation of the DE grade and also of the filter sheet. I show you a capsule in real life. This is how it looks like. You have here a vent, so it's very important that you that the air is completely going out of the capsule before you start the filtration. After the 2-inch capsule, we have the next bigger one, which is the 5-inch capsule. I have it here as well. So it looks similar, a little bit bigger. It also has the air vent. I can show you them later when the presentation is finished in the bigger screen. The 5-inch capsule is mainly used for the evaluation of the 2-inch results and also for small pilot scale. Both, so the 2-inch and the 5-inch, are available as a test kit. So as I said, proof of technology, the 2-inch, evaluation of DE grade and amount, of course, optimization of trials, and it consists of 10 capsules with all 4 DE grades. And the 5-inch test kit consists of 3 capsules and 1 cell pure grade, which is free of choice. And this is the setup in the laboratory scale, what we recommend. So you have the unfiltered liquid with the filtrate, and of course you have to mix it. Then a pump, we recommend here a peristaltic pump, a pressure gauge, and a capsule with the vent. And then you have to collect the filtrate on a scale or with a measuring cylinder. So, and here it is very important that the pump keeps the flux even with increasing pressure. So the pressure can go up to 2.5 bar, and the pump should go to 2.5 bar. Furthermore, the tube connection should be as short as possible, and I will show you that here with this picture. So this is how an ideal setup could look like. the goal of a lab trial is always the same. It's the evaluation of the right DE grade in order to reduce the turbidity. Of course, here you can play a little bit with the DE grade and also the filter sheet, and the optimization of the DE dosage so that you get a high efficiency. So the goal is to reach this green line, an ideal the optimization of pressure and time. Next to the 5-inch capsule, the next one will be the 10-inch capsule. This is the last capsule we have, which is a standalone product. It just needs this filter holder. Yeah, it is also used for small pilot scale. Now I want to move forward with the bags. So they need a stainless support housing. For example, the 12-inch short module, also for pilot scale and small production scale, and the 12-inch single module. Both of them are 12-inch, but this one has much less amount of lenses than this one, and both are available as a ready-to-use version. Sorry, that means that the lenses are increased, so you have a higher number of lenses in a ready-to-use module, because the ready-to-use modules are mainly used for sheet filtration and not alluvial filtration. So for example, when you have a sterile filtration step with depth filter sheets in your pharmaceutical production process. So you don't want to get in touch with your product. I have this module here as well. This is how it looks like. So yeah, I can show it later as well if you wish. And this is the functional principle of it. So you have, as I said, 2 lenses in here, 3 are also possible. And one lenses consists of 2 sheets. So the filter cake is built up on both sides of the lenses. The filtrate is going in the core of the module and goes down, and here's the outlet. So this one would be the outlet, this one is the inlet. And this is how it looks like in real life after filtration. And this is the support housing you need for this small volume module. Sorry, as you can see, it is very a small housing, so you can have it on your table, on your laboratory's table, and yeah. This housing is for the 12-inch normal single module. For all housings, they look the same, just a little bit. The ones are a little bit longer than others. But all of them you can turn 90 degrees in order to remove the modules easily, especially when they are filled with filter cake. They are quite heavy. So, and the 12-inch double module and 16-inch double modules are the last one in this series. So the biggest one is the 16-inch double module, yeah, for the production process. And this is how it looks like in real life. So as you can see on both pictures, the filter cake is built up on each lenses in the same way and looks really good. It still has some space in it, so maybe in this example we could be able to filter a little bit more because the capacity is not reached yet. And this is the housing. As I said, you can turn it to 90 degrees to remove the module. And here we also have a trolley available, yeah, in order that it is easily to discharge. We also do some skids. So as you can see on this picture, here's the module or the housing in the module. We have here a Lavitronics pump, single use, and a control unit. And here is a 5-inch capsule in order to reduce the depth volume. So as soon as you finished your filtration, the bag has still a little bit of liquid in it. And then you can just go out of the inlet and filter the liquid through a 5-inch capsule, and then you can reduce the depth volume. We also can make some skids with many housings in with one control unit. And as soon as you have performed some trials in your lab, you have to calculate a scale up for your production, batch production. And yeah, this is really easy, just one calculation. It is a linear scale up. I already told you something about the powder transfer bags. So there are 3 ways to get the DE powder in your process. First is feeding the filtrate in your bioreactor. Second option is to add the DE to a mixing tank. So if you have your bioreactor and then a mixing tank and then the filter, it is also possible like this. The third option would be that you get the DE as a slurry in your process. So you add the DE with the buffer, for example, and then add it directly in the pipeline before it goes into the filter. A few words about the validation. So to support filter sheets, we recommend here Purifix sheets. These are sheets with low ion and low paraffin containing sheets, and some of them contain also the DE, so the cell pure. Therefore, the pharma grade diatomaceous earth cell pure is already part of the sheets and does not have an additional validation support. Bag material, it consists of double layer PE. The tube consists of APSH, and other plastic parts, like for example the core body of the module or the drainage body, is USP class 6 material, and the powder transfer bags are made of LDPE. We have all validation material available, so just contact us. Now I want to move in some case studies, so 5 different case studies, and I will start with mammalian cell removal for like monoclonal antibody production. So to the background, so you have your mammalian cells like CHO cells, and they produce a monoclonal antibody, and you have a primary and secondary clarification step. As you can see here, after the fermentation, there's a centrifugation, after that a depth filtration, chromatography, virus inactivation, and again a depth filtration step. We can optimize this process as it is. We can use the ready-to-use filter disc, which contains of many lenses, but doesn't have any volume available to build up a filter cake. That's okay if we have a centrifugation step prior to the depth filtration step. Same at the second depth filtration step. Here we can provide a module, for example, with a Purifix HZP sheet. The set indicates that the sheet has a zeta potential, so negatively charged products or molecules like the DNA can be reduced by these sheets. But we want to remove one clarification step. So why not adding an alluvial filtration with our BioSD system? Here we have an example of it. So the cells producing the antibody, cells have approximately 90% viability. We want to harvest it first in a laboratory scale with a 2-inch and then with a 5-inch capsule with a Purifix sheet. The CH09P is quite a coarse sheet, so it is mainly used to hold the filter cake. It has a filtration effect, but the main effect is that the filter cake is not going through. Flux is approximately at 400 to 500 liters per square meter per hour. And here you have a comparison of the different DE amounts. So the blue line is a trial with 10 grams per liter DE. The yellow one, 16.5 grams per liter, and the green one, 13 grams per liter. And as you can see, the green line doesn't go up that much as the other ones. So it goes up, that's normal, but not as fast as the other two. And that's why the same amounts of 13.5 grams per liter were used with the 5-inch capsule. Here you can see the comparison. So the green line again is from the trial with the 2-inch capsule and the blue one with the 5-inch capsule. Of course, the yield is very important. So in order, sometimes you have a product which can bind to the DE. This is possible, but usually, especially in biomanufacturing processes, it's not usual. But it has to be monitored. To conclude this case study, so we had a productive approach with a high flux, 450 liters per square meter per hour, which is quite high. Usually we say for alluvial filtration, you need a minimum flux of 300 to 350 LMH. Yeah, and the throughput was really high with 400 liters per square meter. This is very good. We had a consistent clarification performance. As I already said, these points. The scale up is very easy, and yeah. The next case study would be about the plasmid DNA production. So plasmid DNA is used in the production of viral vectors, as you may know, and here we again have the process with the E. coli fermentation, after that the centrifugation, lysis steps, clarification, endotoxin removal, and purification. And now we can look further to the clarification process. We can do a flocculation and then a depth filtration, or we can do a centrifugation and then a depth filtration. But as you may know, I will recommend alluvial filtration. After that, we have the endotoxin reduction, and also for this we have special sheets. So for the alluvial filtration, we take again the filter disc BioSD, and for the endotoxin removal, we have this Purifix ETR filter sheets. The advantages, it is again a reduction of a process steps, so we don't need the centrifugation step. Therefore, we have a cost reduction. The process time is reduced, and a cost-effective reduction of impurities, especially of endotoxins, with this special filter sheet. It fits to all existing single-use equipment, so it is not a problem to add this Purifix ETR to any of our systems. It is flexible in terms of space for the filter cake, retention rates, and connectors, and stuff like that. Next case study is about viral vector clarification. So as you may know, adeno-associated viruses and lentiviruses are currently being developed for numerous indications in the field of gene therapy. Here again, after the vector amplification, we have a vector expansion, purification, polishing, and fill and finish. For this step, we can use a bioreactor, approximately 550 to 1,000 liter. We have to show cells and do a vector transduction. We have then the lysis and clarification step, which is mainly used with the centrifuge. And after that, again, a depth filtration, which can be made with a ready-to-use system. But again, also here, the alluvial filtration works very well with our BioSD system. This chart indicates a comparison of depth filtration versus filtration with the adding of diatomaceous earth and acid precipitation filtration. So due to adding the filter aid to an unfiltered liquid, as you can see here, the gray line is the unfiltered liquid without any DE, the blue one with DE, so we can filter much more. But when you do an acid treatment and low the pH, we can filter even more. So that's the reason for that is this leads to bigger particles. We have some agglomerates. It is therefore easier to filter. And but here we have to say that before you doing this, you have to check if this influences your gold protein. This is very important. And afterwards, the pH needs to be adjusted again. Last but not least, you can change the filter sheet type in order to get your goal you want to have. So here's an example. We have these cells with a 90% viability. Turbidity is about 300 NTU. We use C1000. This is quite a coarse grade of the cell pure with 25 grams per liter. In trial 1, we used the CH09P, which I already said is quite a coarse filter sheet. In trial 2, we used the CH133P. This is a sterile filter sheet. So you can see it in the retention rate as well. Here we are quite coarse in comparison to this. Average flux was at 648, so you can go with a higher flux when the filter sheet is coarser than with a sterile filter sheet. But even 420 is absolutely fine, especially for an alluvial filtration. It's really good. And the influence, you can see it also not only in the performance, but also in the goal of the quality of your filtrate. So the turbidity is not reduced that much as when you use a sterile filter sheet. Now I want to conclude our system, the FiltroDisc BioSD, with some advantages. It is very flexible. As I said, you can adjust the number of lenses to your process. So this has to be determined in the lab. So the space of filter cake and the filter area can be changed. Connectors can be changed. So all common single-use system connectors can be added. And the retention rate, as I just said a minute ago, by changing the filter sheet, you can remove some finer particles or coarser particles as you desired. We have a very small dead volume by adding a 5-inch capsule for the production scale. The footprint is very small. We just need 1 square meter. High supply safety, so 3 production sites. We have short lead times, normally 6 to 8 weeks. It is widely scalable. And as I said, a well-known technology, especially for plasma fractionation and also in the food and beverage industry, in the beer industry. It removes cells, but also other impurities in one step. And the pH change is possible, but not necessary. Furthermore, we have variable operation possibilities. So you can change from alluvial to sheet filtration. The support housing is exactly the same. The name of the filter disc is just different. So for alluvial filtration, it is BioSD. For sheet filtration, it is ready to use. Also, we have a reduction overall costs and cost of goods. Therefore, reduction of the process time. Reduction of cross-contamination because you don't get in contact with your product. The bag is completely closed. No cleaning validation, therefore you have a reduced downtime, and skid versions are available, including the pump and the control unit. So therefore we can do customized skids. And yeah, we were the first life science industry who can introduce the body feed filtration for midstream application in a single-use format. And one colleague from Merck Serono in France said one day, diatomaceous earth is like the Swiss Army knife, the all-purpose tool for downstream processing. And I think he's quite right. Yeah, I think that's correct. The other comment comes from a famous US biotech company. It's like build your own depth filter because of the constantly new filter surface. And now I come to the end of my presentation, and I
thanks especially to Bettina from Filtrox and also to all the participants. Thanks a lot for being with us, and have a nice Friday. Bye bye.
Thank you as well. Bye.